Okay, so now that we've talked about what coenzymes are generally, we are going to talk about 3 specific coenzymes in our body. So the first one is NAD+, that is used in metabolism. So this stands for nicotinamide adenine dinucleotide. Its structure is over here on the right. So nicotinamide is this part of the molecule here. It's got this, it's called a pyridine ring. It's a nitrogen with kind of that aromatic ring kind of style, and then we have this on the end. This nicotinamide is derived from niacin, which is vitamin B3. So that's why it's one of those important vitamins that we need in our bodies, is so that we have access to this part of the molecule, or this part of this coenzyme, to allow our processes to work fully and correctly. So the other parts of this are ADP. So over here you see we just have the same ADP, 2 phosphates because we have that D there. And then at the end of that phosphate is where we connect to another ribose and then the nicotinamide. So this whole part, the yellow and I guess salmon color here, all of this stays the same in this reaction, just so you know. That's why it kind of disappears here. So what this does, let's do a new color. I like fun colors. Is that it participates in oxidation reactions that produce carbon-oxygen double bonds. So specifically, that's too dark. Let's just circle it a bunch. Carbon-oxygen double bonds. So NAD+, anytime you go from a carbon-oxygen single bond to a carbon-oxygen double bond, you're going to be using this coenzyme to accept the hydrogen. So let's look up here on the top. So we see this structure here for NAD+. See how there's a plus in the oxidized form? That's because there's a positive charge here on the nitrogen. Then when you add in H2 plus 2 electrons, you can kind of think of this as you have an H minus and an H plus. The minus is going to be a lone pair of electrons. That's where those 2 electrons are going. And then this H minus is going to add in right here. These electrons will move. These electrons are going to go onto the nitrogen to give it a lone pair here. So that H minus ended up here, and then we just have an H plus floating around as well. But NAD plus is going to accept those hydrogens. This it says it's kind of floating. To be honest, it's actually complex, but it's just not covalently bonded. So NAD plus is reduced in order to form its reduced form that looks like this over here. So where this is used, let's look at this reaction on the bottom. So we have ethanol in this case with NAD plus and an alcohol dehydrogenase. Let's write alcohol just for clarity, because it's going to be pretty much any alcohol. We remove H2 from the alcohol. So that's what these bolded hydrogens are for. These 2 hydrogens are going to be removed and in order to form that carbonyl. So now we have an aldehyde. And NADH, the reduced form of NAD plus H plus. So those hydrogens are the same hydrogens that we had in our alcohol. Okay, the next coenzyme we're going to look at is FAD. So FAD stands for flavin adenine dinucleotide. So looking at the structure, we have flavin up here in purple. This is made, or a portion, it just changes the name a little bit, but when you attach it, but it contains riboflavin, which is just this part on the top. And that is vitamin B2. So that's why we need vitamin B2 in our diets. And it does the same kind of thing where it accepts 2 hydrogens and 2 electrons, specifically in the processes that form carbon-carbon double bonds. So in this case, you see these arrows here. One hydrogen goes onto here, one hydrogen goes onto this guy. So now we have let's circle them. Let's do a different color though. We'll add these hydrogens on. You also see the double bonds move around a little bit. But again, FAD is going to accept these two hydrogens specifically in reactions to make carbon-carbon double bonds. And then we call that FADH2 in its reduced form, FADH2. Okay, so let's look here. For example, if we had something like this, which is called succinate, sorry, there's succinamine, and that's what I was trying to get to, but succinate, if we have a succinate dehydrogenase, succinate dehydrogenase, and your FAD coenzyme, then you can catalyze the reaction to remove two of the hydrogens, the ones in bold, and make that double bond, and that reduces your coenzyme so that you get your two hydrogens out in your reduced form of the coenzyme. The last one we're going to talk about is coenzyme A, or CoA. This is a coenzyme that is needed for acyl transfer reactions. We'll talk about these more a little bit later and to why they are important in our bodies. But overall, it's a transfer reaction. We're taking an acetyl group and moving it to a different place. This is the structure of coenzyme A. We showed it before. The important part here to notice is that it has that thiol on the end. The rest of it is kind of doesn't do too much in the reaction, but it's important structurally for how our body perceives everything. But this pantothenic acid, so this portion in the middle, about here over to here, all this little chunk is called pantothenic acid. Pantothenic acid is vitamin B5. Then we also have an ADP over here, and then aminoethane thiol on the end over here to make it up, finish it up. So this is going to be abbreviated CoA, big C, little o, big A. Important that it's in that particular order or particular capitalization. Remember, it is a thioester. That is important. It is more reactive than esters. I'm sorry. More reactive, yes. Okay, well, so the reaction that acetyl or coenzyme A catalyzes or helps as a coenzyme for is when you take an acetyl group, so this whole group here, and so pretend like there's an R here, so there's more of this molecule, there's more to it. Coenzyme A and your enzyme, you're going to make a bond in between your carbon and your sulfur, and you make a thioester. That's what this thioester is here. So this group is called a thioester. It is pretty reactive. It's more reactive than typical esters that we've seen before. So it's a transfer reagent. It adds on, and then it can react again easily and transfer that back to something else. So you'll see this one a little bit later and see why it's important in our citric acid cycle. And this, it seems kind of weird at the moment, but it is an important reaction. But overall, remember that it is going to catalyze our acyl transfers. Okay, our last one is our learning checks. So which of the following coenzymes best match with the description? So we have 5 coenzyme names in both their oxidized and reduced forms for almost all of them. And then we also have 5 different examples here. So what is the coenzyme used in oxidation of carbon-oxygen bonds? So that is our first example that we did. That was NAD+. So this is the coenzyme used, meaning it hasn't been oxidized or it hasn't been reduced yet. Okay, then we have B, the reduced form of flavin adenine dinucleotide. So flavin adenine dinucleotide, we see that flavin, that's the important part. That's the FAD, but it's going to be the reduced form. So we're going to have hydrogen attached. So that would be FADH2 with our 2 hydrogens on their reduced form. C, used to transfer acetyl groups. We just talked about that one. That one is CoA, coenzyme A. We have D, oxidized form of flavin adenine dinucleotide. So this is very similar to part B, right? But now it's the oxidized version. We don't have those hydrogens on there, so it's just FAD. And then the last one, the coenzyme after C double bond O bond formation. So after we form that carbon oxygen double bond, we removed those hydrogens from NAD plus, or removed those hydrogens from the alcohol, added them to NAD plus. So now we have NADH plus H plus out of that reaction.